Hey guys, welcome back. This is chapter 12, so we're moving right along in our lessons. So chapter 12 is on gene mutations. So mutations are changes in the DNA, and we're looking at how they affect genes. Um, a lot of the sections are kind of short, so this presentation is actually going to involve sections 1, 2, and 3 of the notes. So, without further ado, let us get going, shall we? All right. So, section 1, the nature of mutations. So, as we said, a mutation is a change to the actual DNA within a gene. So, remember, you have your DNA, and it's made up of these things called nucleotides. And nucleotides involve a sugar, phosphate, and base. So what a mutation is, it's a change in the bases. So whether you're switching one base for another, and if you look at the picture at the bottom, here we're going through replication, and the original gene or DNA has, where am I at? So they're showing you the original right here. And here we're seeing where it makes the copy. So green pairs with purple. Now down here, instead of making a new purple base, it created a change. Yeah. So here in the original, there's the purple, remember, complementary. And we would expect there to be a green one here, but instead of green, it's a yellow base. So we have a change in the bases. Now that can lead to different expressions or phenotypes. That's actually what they believe was what happened with blue eyes. About give or take 10,000 years ago, there was a mutation in the DNA and it led to a different coloration by changing the depositing of pigments in the eye. So types of mutations. So this is just very, very broad and basic. We're actually going to get to the more specific types in a little bit. Mutations include single base changes. So that means changing one base for another. So for instance, if you have a guanine, changing it to a cytosine or thymine or adenine. Deletions means that you are losing part of the DNA. So it actually gets taken out and the remaining gets put together. Additions, adding extra stuff to the DNA, so adding extra bases, or move sequences. So what if the entire gene kind of moved around, or sections of the gene got moved around and rearranged? Now, specifically when we talk about mutations, we're more so going to talk about the ones that occur in the part of the DNA that codes for proteins, because that's where we're going to see most of the effects from. If it's in any of those signs or lines or repeating sequences, we're not going to see any effect from them. But if it's in a section that is an actual gene and codes for a protein, it could potentially have no effect. It could potentially have a lethal effect. And there's a whole wide range in between there. You can also divide them up based on kind of where they occur. So germline mutations are ones that can be inherited, meaning that they affect the gametes, so the sex cells, the egg and sperm. Somatic mutations cannot be inherited. Now, we give cancer as a particular example of that one. There is genetic predisposition, so a gene or genes that can affect your likelihood of getting cancer. But specifically, in terms of somatic mutations, I'm talking about specific cancers. For instance, if I go out in the sun, I know there's not a whole lot of sun today, but I go out in the sun, I never wear sunblock, and I, get exposed to lots and lots of UV rays, and maybe I got a mutation right here in my skin cells there that changes and mutates and becomes skin cancer. Now, that's not something I can pass on. what would happen is I could get it treated, removed, but this cell right here is not going to affect the DNA in my egg cells. So that's what we're talking about with these somatic mutations. They occur in cells of your body that are already formed, as opposed to germline ones, which occur in cells such as that are important for reproduction, such as either the predecessor cells, the diploid cells that will go through meiosis to make the sperm, or the egg cells once they're already formed in the female. All right, let's see. So here's a specific example of a mutation. It's called CCR5, and it is homozygous recessive, meaning you need 2 of the copies of this changed allele in order to express this trait or lack of expression. It's very rare. It only occurs in less than 1% of the population. Now, the interesting thing about this is it's a defect that encodes protein on the cell surface that HIV must bond to, so the individual does not get HIV. So what we're saying is, if you look at the picture, there are receptors on your cell surface. And oftentimes, the HIV virus can actually attach to those receptors, and that's what allows them to gain access to the cell and allows them to enter the cell, hijack it, use it to create more of the virus. But someone with this particular mutation, with CCR5, it actually affects those receptors. So those receptors are no longer able to bind HIV, which means that people with this mutation, even if they're exposed to HIV, they cannot contract it. So they cannot ever actually contract HIV, which means they can never get AIDS. Now, a lot of times when we talk about mutations, we talk about them in terms of negative effects, but this is just showing you that not all mutations necessarily are bad. Mutations can be good. Actually, you can classify them as good, bad, or neutral. Good, good meaning they help an organism function better or survive better. Bad meaning they affect an individual's chances of survival in a negative way. And neutral meaning they don't affect your chances of survival. For instance, blue eyes. It's a neutral mutation. It doesn't help me live better. It's not like it improves my eyesight. And it doesn't affect my chances of living. It's not going to cause me to get sick and die or damage my eyesight just because I have blue eyes. All right. Now let's move on to section 2. So we're going to talk about mutations. Specifically, we're going to look at a couple of disorders. We're going to look at disorders of the beta-globin gene, as well as disorders of collagen. Now, we've talked about globin genes before. Remember when we were talking about hemoglobins and how they can be affected? So we're going to actually talk specifically about 2 mutations in the gene for making the beta subunit of hemoglobin. First one is called sickle cell disease. Now, in the past, a lot of times it's been referred to as sickle cell anemia, but now that they know more about it, they're trying to move away from the term anemia and go towards disorder or disease, because they found out that it actually is caused by a recessive mutation. Now, when we talk about these things as mutations, we're not saying that someone who has sickle cell necessarily, it's not that their cells went through that mutation. But what we're saying is that somewhere along the line, the ancestral line, they're related at some point to someone who had the initial mutation, So for instance, I didn't just randomly somehow mutate and get blue eyes. If we look along my my ancestry history, 10,000 years ago, I'm related to that person who first had the mutation. Now, it's still caused by that mutation, which is why we still call it a mutation, but now it's more normal. And everyone in the world who has blue eyes, we are all related to that common ancestor who first had the mutation 10,000 years ago. Now, the reason why I brought that one up is because I don't exactly know when sickle cell disease, when the mutation occurred, whereas I know when the blue-eyed one occurs. So anyway, sickle cell disease is recessive, so that means you need 2 copies of the recessive allele in order to express it, and is caused by a single nucleotide substitution, meaning that it's one base gets substituted for another. So if you look here, in the gene for the beta subunit, the beta-globin subunit, there's a codon that is normally CTC. So in the DNA, it's cytosine, thymine, cytosine. You go through that, you do transcription, that'd be GAG, and that would code for glutamic acid. So that glutamic acid would be found in a particular location in the beta subunit. However, in people with sickle cell, they don't have the thymine there. Instead, they have an adenine present in place of it. So in their DNA, the codon says CAC, cytosine, adenine, cytosine. And that turns the mRNA into GUG, which codes for valine. Now, when that codes for that different amino acid, even though it's just one amino acid that's different, it's going to affect how the amino acid chain can actually fold up into a 3D structure. And because it affects how it folds up, it will have an effect on the expression of the beta-globin subunit. So this is kind of showing you a little bit about what we're talking about. So normally in normal blood cells, red blood cells, you can kind of see they're this donut shape. This is if you cut one in half. It's kind of puffed on the outside and thinner on the inside. It looks like a little round flexible donut, and it can easily go through the different blood vessels. However, with sickle cell, the blood doesn't make the normal shape. Instead of being round and puffy, it's kind of elongated and curved, kind of like a sickle or a crescent, which is why it's called sickle cell. And because of the elongated curved shape, it's more likely to get caught in some of the smaller blood vessels. And once one of them gets caught, it's going to start blocking that entrance, which means more of them are going to get caught, and it can actually lead to blood clots in the vascular system. Now, that is definitely a negative. It could lead to a lot of pain, a lot of other physical troubles, such as, depending on where the blood clots are more likely to occur. I've heard of massive leg pains. I've heard of headaches. I've heard of people needing surgeries to kind of fix these things. So it can be very, very negative. I do want to say, though, one more thing is that it is negative, and I'm not saying that it's good. I don't have it, so I can't really speak too truly on this. But one of the thoughts as to why this mutation might have occurred is because when the blood has that elongated curved shape, it actually makes it so that the parasites or the disease of malaria can't attach into the blood. So people with sickle cell are also immune to malaria. So it's thought that maybe that was one reason why there is mutation. It affects the blood flow and how it moves through the vascular system, but it also could help individuals who are in malaria afflicted countries to better survive from that particular disease. All right, so next beta-globin disorder is called beta thalassemia. So what this does is it actually creates excess iron in the blood. And the reason for this is because the beta-globin subunit is actually either absent altogether or it's very, very scarce. So there's not as much of it to make the red blood cells and the hemoglobin properly. So you either don't have the beta-globin or it is absent. Oh, sorry, that's the same thing. Or it's just there's not much of it being made. As a result of this, the symptoms, it actually starts. So remember how we talked about the 3 types of hemoglobin, the embryonic, the fetal, and the adult? So after someone is born and they switch from the fetal hemoglobin to the adult hemoglobin, there's still some fetal hemoglobin coursing through their system. The genes are not going to make any more of it. They're going to start making the adult hemoglobin, but the existing fetal hemoglobin in the blood cells are still there. And eventually they do get worn out, broken down, and replaced by the adult hemoglobin. And that's actually when the symptoms of beta thalassemia become seen. When the fetal hemoglobin are depleted, then you start to see the effects of beta thalassemia, because if you remember, like way back when we talked about this, the fetal hemoglobin actually doesn't have the beta subunits. It has alpha and delta, I believe, whereas the adult hemoglobin has alpha and beta. Well, once we start trying to make these adult subunit or adult hemoglobin, and we cannot make the correct subunits for it, it starts having pretty large massive effects. For instance, the lack of the beta subunits, we already said it causes an increase in iron. Now that iron can get deposited in the different organs and absorbed by the organs like the heart, the liver, different endocrine glands. And since the iron's building up in there, it actually starts to cause damage to the organs. Plus, without the beta subunit, we're not able to transport oxygen as well either. So your organs start to have less oxygen supply, so they're depleting their oxygen supply. Plus, we're getting a buildup of iron in them. Now, iron is a heavy metal, and it's needed in small amounts, but in large amounts, it actually becomes toxic. And that's why it starts to destroy your certain organs that are very much blood enriched. They have found that it does, because it changes the shape of the blood, just like how sickle cell does, that it also does protect against malaria. So once again, there's this other benefit. There is this benefit of an otherwise horrible disorder that could have some kind of evolutionary benefit, because they have found that it typically is found in people from certain ethnic backgrounds. So people whose backgrounds include Italy, different Mediterranean countries, North Africa, the Middle East, India, South and Central Asia, because that's where a lot of, at one point, not as much maybe today, but at one point, malaria was very prevalent in many of those areas. It still is today. And so that's one of the reasons why it's thought this mutation could have occurred, because the individuals from those areas were exposed to malaria, and maybe there's a mutation that evolved that helped them to not contract the disease. However, it does have this negative effect of causing beta thalassemia instead. All right, so again, here's a picture of the blood cells. In normal red blood cells, we got those round little donut puffs. And in the beta thalassemia, they're very flat, like we saw with the sickle cell, but instead of being curved, they're like flat sheets. Now, just like before, they can, because of the change in shape, they can also lead to an increased risk of blood clots. All right, so let's talk about collagen disorders. So collagen is a symmetrical protein that's found in tissues, blood, cartilage, skin, ligaments, and tendons, blood vessel linings. It's also what separates cell types in tissues. So it is a pretty versatile protein. It gives a lot of our tissues strength and flexibility. Now, when you have a disorder in the collagen and it's not as or it's not made as well, or it's not made in the normal way, then that can cause it to either be too flexible and not strong enough, or too rigid, so it can't really flex. It could also affect the strength, or we already mentioned how it can affect the strength. So symptoms of some do include very stretchy skin, as we see in the picture on the bottom left. So there's this guy here, he has super stretchy skin because, again, in this case, the collagen is too flexible. So like us, we can't really pull our skin that far away because the collagen still maintains some rigidity and strength to keep all the tissues together. But in that case, it's much, much too flexible, and it actually loses a lot of its supportive nature. Aortic aneurysm, so that's if the aorta is the main artery that leaves the heart. And what happens there is, if it can't be flexible enough to, expand and contract in order to pump the blood, it can't do that. It can actually burst and lead to an aneurysm, which means that the blood vessel has burst and now you're essentially internally bleeding to death. Two of the ones that we are going to mention are L-ports, which causes deafness and inflamed kidneys. We'll talk about that, as well as Marfan Syndrome. I do want to talk just briefly about these two pictures here. So the picture in the hand in the upper left and the feet in the bottom middle. That's just, again, showing you the excess flexibility that can occur with certain collagen disorders. All right, let's move on. So L-port syndrome. Now, what this does is, again, it affects how the collagen is made. It is a protein. We have genes to make it. And what happens is, if you kind of look here at the bottom left, within the kidneys, you have this layer of connective tissue in between, or within the blood vessels, that separates the blood and the urine. Now, what happens is it gets thick and more rigid, so it can't really allow for as much of the absorption, because how our kidneys work is we filter the blood in the kidneys, and we can take excess water and filter it through that collagen layer to the kidneys, so where it can be then sent to the bladder. It can also filter out ions, salts, things like that. But in this case, it can't filter it as well. So we do notice that there can be a loss of kidney function. The kidneys will essentially start failing. And even with other things such as hearing loss and eye abnormalities. So there's a lot of connective tissue around your eyes and within your ears. And this doesn't show the eyes, this is just showing ears. So what happens is this Reissner's membrane is around here within your ear. Now it can lead to hearing loss because, again, it's a flexible membrane of protein that helps to transport the sound waves. But if it thickens up, if it becomes too rigid, then it can't actually absorb the sound waves and pass them on. And we can have similar problems with the eyes in terms of the eyes, the proteins, the collagen in it doesn't work as well for the absorption of light or adjusting the lens so we can focus on things. So this one we tend to see a lot more of thickening and the protein being more rigid than it should. And then here we have Marfan syndrome. So this affects the body's connective tissues that hold everything together. So as opposed to looking at very particular locations, like very specific areas of protein, of this collagen protein, Marfan syndrome, it more so affects the connective tissue as opposed to specific membranes and things like that. So because it affects how the tissues and the organs are all held together, we do tend to see some very striking, noticeable physiological or anatomical differences, such as the elongated hands and fingers and toes on the foot. You also notice that there's elongated limbs and very, very pronounced rib cage and sternum. A lot of that is just because of how the connective tissue isn't holding things together properly, so they kind of stretch out a little bit more. Even if you look at this hand down here, we already mentioned that it's a little bit too flexible in this case, as opposed to L-port where it was too rigid. In Marfan, it tends to be excessively rigid, which can then affect the strength of the structures. If you look at this picture here, here's the thumb here. That's the pinky. That's this finger. So imagine like keeping the rest of your hand straight and just bending that so it comes all the way through. That's how much flexibility that person has in their hands. And this might sound cool in terms of, you know, hey, you got longer limbs and you got increased flexibility. Yeah, but like we already said, it can affect the strength of it. The collagen has a very delicate balance of flexibility and strength and stability. If you're too flexible, it loses that strength, which is why there's oftentimes heart problems. There's a lot of collagen around your heart because it has to beat and flex. So it has to contract and constrict and beat in and out, as well as the blood vessels coming out of it, the aorta, as well as the vena cava going into it. So it has to be flexible yet strong. And a lot of times what we see with Marfan syndrome is you got the extra flexibility, but it actually decreases the strength and causes more heart problems because of that. All right, section 3, allelic disorders. So we're only going to really mention one of these, and that's presenilin. And this is a particular protein that affects specifically receptors of the Golgi apparatus. So in the in membranes, whether it's cell membrane, nuclear membrane, the Golgi membrane, there are receptors on there, and those receptors are made up of proteins. And this is a particular protein receptor that is located on the Golgi apparatus. Remember, that is our shipping organ now. It packages proteins, lipids, etc., to be sent out of the cell. And what they notice about this particular one, if you have the presenilin protein disorder, one of the common things that it causes is early onset Alzheimer's. So Alzheimer's is a form of dementia, and more often than not, it happens later in life, but this can have it starting earlier in like the 30s or 40s as opposed to the 60s or 70s or 80s. And the reason why of this is because there's an abnormal amount of amyloid built up in the brain. So specifically, this is affecting the Golgi apparatuses in your brain cells. Now it's affecting ones in other places as well, but it's because of the ones in your brain cells that we have the Alzheimer's, the early onset Alzheimer's. So the presenilin protein disorders, the receptors cannot process or recognize and process the amyloid. So that means if it can't process it, it can't get it ready to ship outside the cell, which means it stays in the cell. So we end up seeing these large amounts of buildup of amyloid in the cells within the brain. And it's this buildup that leads to the early onset of dementia. Most people, because the Golgi apparatus and its the presenilin receptors can detect, receive the amyloid, package it, send it out. Even with people who might get Alzheimer's later on, they don't have an issue with that until much later in life. But because of this starting up right away, the buildup occurs sooner in life, which is why it causes Alzheimer's sooner in life. All right, so I'm not going to ask you to really completely understand this. This is just if you wanted to know a little bit more about the biochemical pathways that's going on that leads to this. You're not going to be tested on this. It's just, I know sometimes kids in microbio, or if they're in RP or if they're an AP bio, might be more interested. I just kind of wanted to show this to you. All right. And that is it for sections 1, 2, and 3. So we got lots that we covered. We do have 2 more video presentations for this chapter, which will actually talk about the types of mutations, how they occur, what kind of effects they can have. And yeah, so hope this helps. Hope you enjoyed yourself. Hope you learned some good things. If you have any questions, hit me up in our Google Classroom or through email. And yeah, just stay awesome, you guys. Stay healthy, stay great, and I'll talk to you later. All right, bye-bye.